Good afternoon, everybody, and welcome to the latest in the Verimed webinar series. This afternoon, we have Clare Newall talking on the early phase PK study. Clare's currently Statistics Manager at Verimed, obviously based in Twickenham. She previously worked for over 11 years in early phase biostatistics at a global CRO with a significant focus on pharmacokinetic studies. So a couple of housekeeping points. If you haven't attended a webinar with us before, you'll notice that you're automatically muted. If you want to ask any questions throughout the session, we'll be presenting those to Clare at the end in the Q&A session. And you can do that by typing your question into the questions box that you see on your GoToWebinar toolbar. And I'll be presenting those to Clare at the end of the session. So without any further ado, I will pass you on to Clare. Thanks, Clare.
Thank you. Hi, welcome to this webinar, which aims to provide a very practical insight into some of the challenges and operational considerations to be taken into account when planning and reporting the results of a PK study. This is not intended to be a theoretical presentation, but I will include a brief overview and refresher of some key PK concepts and follow with considerations for the statistical analysis plan and regulatory guidance. I will also be discussing some common data issues, as well as providing some practical and logistical insight into planning and conducting the PK study. And as Louise says, I'll be happy to take any questions at the end. Pharmacokinetics is the study of the absorption, distribution, metabolism, and excretion of a specific drug, known as ADME. It is a key component of early phase studies, including first-in-human, bioequivalence, food effect, drug-drug interaction, and other studies in specific populations. It is also a regulatory requirement, is used to assist in formulation, development, and optimization, as well as contributing to drug labeling information. In terms of assessing pharmacokinetics in a clinical study, typically, drug is administered at time X, and a series of blood samples are then taken at pre-specified time points, which are subsequently analysed in order to provide a profile of the drug concentration over time. The choice of time points is usually made based on preclinical knowledge or knowledge gained from other clinical studies regarding the expected absorption and elimination rates of the drug. There are usually more frequent samples around the expected time of the maximum concentration in order to ensure that the maximum exposure is determined as accurately as possible. The analysis of PK data may be performed using compartmental or non-compartmental methods, and population PK models may also be used. The majority of early phase clinical pharmacology studies tend to use non-compartmental analysis, which is a model-independent approach that assumes that the drug follows first-order kinetics. In most protocols, this is usually referred to as NCA and is performed using specialized software, typically Win-Non-Lin, or Phoenix as it's often now referred to. Very briefly, I just wanted to refresh some terminology as I will be using these again in later slides. So this slide just provides a very simple representation of a concentration time profile following a single oral dose, plasma concentration on the y-axis and time on the x-axis. Following dosing, the drug concentration increases to a maximum Cmax, which occurs at a specific time, which is then termed Tmax. It should be remembered that Cmax and Tmax are entirely reliant on taking samples at appropriate times following dosing, and that time window deviations for samples planned around the expected time of Cmax may result in the true Cmax being missed and therefore underestimated. Clast is the time of the last quantifiable concentration. The AUC 0 to T is the area under the curve from time 0 to the time of the last quantifiable concentration, and lambda Z is the elimination rate constant from which the drug half-life is then calculated. One important concept that needs to be taken into account during the analysis is the lower limit of quantification. And this is essentially the lowest concentration that can be measured by a specific assay. As the drug is eliminated from the blood, the concentration measured in the sample decreases until at some point, as you can see on the graph on the left-hand side, the assay will no longer be sensitive enough to detect the drug in the sample. The point at 8 hours post-dose is the time of the last quantifiable concentration, which is then T-last, and the area under the curve from time 0 to time T-last is in this case the time from time 0 to 8 hours. We can also calculate the area under the curve from time 0 to infinity by extrapolating beyond the last quantifiable concentration using lambda z. The reason that this is of importance is that we need to have rules for how to handle these so-called BLQ measurements in terms of both calculation of the PK parameters and the reporting in table figures and listings. These rules should be included in the statistical analysis plan and may often be different for the calculation of PK parameters and the calculation of descriptive statistics. In other words, there may be more than one imputation rule required for measurements that are below the limit of quantification. So this is my last theoretical slide before I move on to the statistical analysis plan and provides a very brief overview of the PK parameters that may be measured in the urine. Urine PK is a common endpoint in phase 1 studies and allows the assessment of the amount of drug that is excreted in the urine, which therefore reflects the clearance of the drug via the kidneys. Typically, several samples are collected. Firstly, a pre-dose sample, which is essentially just a spot sample rather than a discrete collection. Then, following dosing, urine is collected over subsequent intervals, for example, 0 to 2, 2 to 4 hours, and so on up to 48 hours post-dose, for example. From each of these intervals, the volume of urine is measured together with the concentration of the drug, and these are then multiplied to get the overall amount of drug excreted in that interval, which is then known as AE T1 to T2. The amount excreted is then summed over each collection interval in order to get the cumulative and total amount excreted, which can also then be expressed as a fraction of the total dose administered, known as SE%. Often we create plots showing the cumulative amount excreted over time after dosing, where the cumulative amount is calculated for each collection interval and then plotted against the midpoint of that interval. So 2 important things to note about urine PK is that, again, we might have BLQ concentrations, so the SAP should include rules about how to handle these. And secondly, generally speaking, there is no modelling required for urine PK data. So it is not really necessary for these parameters to be calculated by a PK scientist using specialised software such as Win-non-Lin. They could just be calculated by a statistical programmer in SAS, for example. In my experience, the responsibility for this piece can vary depending on who is calculating the PK parameters and the relevant SOPs. Some will stipulate that all PK parameters should be calculated within a PK department, and some will allow the urine PK parameters to be calculated by the statistical programming team. And the reason that I'm mentioning this here is that there is often some confusion over the responsibility for this part, so it is important to ensure it is clear at the outset who is doing this. You don't want to make the assumption that the PK scientist is doing it and then realise when you receive the PK parameters that the urine data are not included. So now I'm going to move on to the statistical analysis plan, and the next 2 slides look at data collection. I like to include in the SAP exactly what data is being collected and then how this will be used in the analysis. The protocol does not always include the full details which may be of importance for the SAP, and this information may only be obtained from the CRF. Dosing information is key. The time of dosing and amount of drug administered should always be reported, although the format of this may depend on the formulation being administered. For example, for an infusion, it might be the volume that is reported, and we would also need both the start and stop time, as the duration is used in the calculation of the PK parameters. For a drug-drug interaction study, we might calculate PK parameters for more than one analyte or drug, and in this case, we need to ensure that we report the time of dosing for both compounds so that actual sampling times can be calculated correctly. One very important thing to note at this point is that it is very common in a first-in-human dose escalation study to modify the planned dose levels. So we might have planned dose levels in the protocol of 100, 200, 300 and 400 milligrams, for example, but actually during the study we modify the highest dose level, for example, to 350 milligrams. And it is very important to ensure that any modification of dose levels is communicated from the project manager back to data management and biostatistics, such that we can ensure we have the correct dose levels in the database and then use these in the subsequent analysis. PK sampling times are also important, and I have a couple of slides on this later, so I won't go into this more now. For a food effect study, we would typically have the start and stop time of the high-fat breakfast, together with an indicator of the completeness of the meal, which might be in the form of a percentage of the meal consumed. And this can then be used as a basis for excluding non-compliant subjects from the PK analysis. For adverse events and concomitant medication data, we would always expect the start and stop time. In later phase studies, times are not usually collected, but for first-in-human studies, where subjects are in-house, it is usual to collect the times, as these might be important in determining the effect of any events on the PK data. For example, if vomiting occurs after oral dosing, it might not be considered important if this occurred at 10 hours post-dose, compared to, say, the same event at 1 hour post-dose. For drug-drug interaction studies, it is typical to collect relationship data for each specific investigational product. So this should be taken into account when setting up adverse event tables, as we might have two sets of causality data, one for each drug in the study. For other types of studies, there may be other data of relevance. For example, in a PK study in patients with renal impairment, the glomerular filtration rate is used to quantify the severity of patients. Similarly, in hepatic impairment studies, again, some assessment of severity is needed in order to categorize patients, and here it is usually the Child-Pugh classification. And there may be some collection of blood samples, for example, for genetic analyses. We might be interested in protein binding, as well as measuring other relevant safety laboratory variables, such as prothrombin time, that may not form part of the usual safety laboratory panel in a first-in-human study. Both FDA and EMA guidelines provide details regarding other information that is required for these types of studies and how this should be used in the analysis. So it is important to consult this when writing the SAP. For example, FDA guidance advocates the modelling of the relationship between PK parameters, such as renal clearance, and the glomerular filtration rate, in order to try and quantify the relationship between the degree of renal impairment and the impact on the PK of a specific drug. So I would now like to come back to the issue of sampling schedules, which can often be confusing in the protocol, both in terms of the reference point for samples and the time points to be included in the calculation of any PK parameters. So for example, for an infusion, it should be very clear whether time points are relative to the start or end of dosing. I've seen protocols where it states samples will be taken at pre-dose and specific time points post-dose, but it is not clear whether we mean after the start of the infusion or after the end of the infusion. If we mean after the end of a 1 hour infusion, the 2 hour post-infusion time point is actually the same as 3 hours post-dose in terms of calculating PK parameters, as these are always based on the start time of the infusion period. Secondly, it is important to review how samples are being labelled in the database. For example, the 24-hour post-dose time point on day 1 might actually be labelled in the database as day 2 pre-dose in a multiple dosing study. And again, as I mentioned previously, in first-in-human dose escalation studies, there is often some flexibility in the protocol, and this time there may be flexibility around the timing of samples, such that these may be modified from one cohort to the next, provided that the total amount of blood taken is not altered. This can be very tricky to keep track of in terms of what is expected for each cohort. So it is important to ensure that there is an appropriate mechanism of feeding this information back to data management and biostatistics so that time points don't get inadvertently dropped from the data set and the analysis. Finally, it is useful to clarify in the SAP over which time period the PK profile is being calculated, as this informs how the actual relative sampling times will be calculated and how the data will then appear in the listings and tables. It might also be necessary to duplicate data across multiple rows of the analysis data set, such that the same time points may have different labels depending on how it is being used in the analysis. And I have some examples of this on the next slide. So in this example from a multiple dosing study, the 24-hour time point here is labelled as pre-dose day 2 and would need to be renamed back to 24 hours post-dose if we wanted to calculate a PK profile over 24 hours from dosing on day 1. So in this case, we would relabel the time point back to 24 hours, and then we would calculate the actual relative time to dosing on day 1, rather than dosing on day 2, as it was previously. The situation can be slightly more complicated for twice daily dosing or other regimens. In this example, dosing is twice daily, 12 hours apart. The protocol shows the time points on day 1, all relative to the first dose, and this has actually been taken from a study that I have previously worked on. So in this case, the 12-hour time point is also the pre-dose time point for the second dose, and the 14-hour time point is the 2-hour post-dose time point for the second dose, and so on. This slide shows how this data could then be handled within the analysis data set, such that we duplicate certain records depending on how they are going to be used in the analysis. So if we take the 12-hour post-dose time point, this is also going to serve as the pre-dose time point for the second dose, and it might also be used in an analysis or graphical representation of the trough samples in order to assess achievement of steady state. So here we create a new record with the same concentration data, but now we label this as pre-dose with nominal time of 0, and then we can use flags within the dataset to select the appropriate record depending on the analysis. For the second dose, assuming we again wanted to calculate the PK parameters, we would relabel all the time points so that they were relative to the second dose. Thus 14 becomes 2, 16 becomes 4, and so on up to 12 hours post-dose. Similarly, we might generate a new record for the 12-hour post-dose sample while retaining the trough sample record for pre-dose day 2. The SAP should always include details regarding handling of BLQ values, and as I mentioned previously, this may require different rules for calculation of PK parameters versus rules for calculating descriptive statistics of concentrations at each time point. And there may also be other rules for how you present individual figures, as well as for urine PK parameter calculations. It is important to ensure that the rules for each of these are clearly stated in the SAP, and then these can be accommodated in the analysis dataset by inserting new records as required and using appropriate analysis flags. Typically, in the listings, the data are presented as received from the bioanalytical lab without imputation. So BLQ values will be displayed as BLQ in the listings. Then there will be rules for calculation of PK parameters. For example, pre-dose values might be substituted with 0. Post-dose BLQ values between two quantifiable values might be set to 0. And terminal BLQ values, which are those at the end of the profile, are usually regarded as missing. For descriptive statistics of concentrations at each time point, we might substitute with 0 or with half the lower limit of quantification, for example. The rules for substitution are usually at the discretion of the PK scientist. They might be based on previous studies with the same drug, on SOPs of either the CRO or the sponsor, and there are many different variations that I have seen used. Thus, it is important to ensure that the SAP is very clear about how these should be handled in order to avoid rework later on. So this slide shows a very simple example of substituting BLQ values in an analysis data set. For this subject, the pre-dose and 48-hour post-dose values were BLQ. We have three columns on the right-hand side, which are analysis flag columns for listings, tables of descriptive statistics of concentrations at each time point, and finally, the win-non-lin analysis flag, which is for the substitutions that will be used to calculate the PK parameters. So, for the pre-dose time point, we substitute with 0 for both descriptive statistics and the calculation of the PK parameters. So we only need to create one additional record with 0 imputed, and this is then used for the tables and for the win-non-lin analysis. The original value here as A-val-C, as BLQ in A-val-C is then used for the listings. For the 48-hour time point, we again insert a new record for the descriptive statistics with the analysis 2 flag set to Y. This is again imputed as 0 in this example. Note that in the typical case where terminal BLQ values are regarded as missing for the calculation of the PK parameters, we do not need to create an additional row here, as this would be blank for A-val, and therefore would anyway get ignored by win-non-lin, as if effectively it wasn't there. And this principle can be followed for more complicated data and rules, and additional analysis flags may also be required. The SAP should always include a complete list of the required PK parameters. The key PK parameters are usually specified in the protocol. However, typically this may omit some of the variables that may be required to help interpret the data. These so-called diagnostic PK parameters are usually required to be listed and help inform the validity of the modelling, but these are often not stipulated in the protocol. And these may include the percentage of area extrapolated in the estimation of AUC to infinity, the R-squared, and lower and upper time points used for the estimation of the terminal elimination half-life. Thus, it is important to ensure that the SAP includes all the variables that will be included in the TFLs. There's nothing worse than getting to the reporting phase and then a reviewer asks for the R-squared values, and these have been dropped from the PK parameter file coming from win-non-lin because they were not specified in the SAP. As I already mentioned, the SAP should have clear rules for handling BLQ values, as well as stating the reference time point for the calculation of actual sampling times. For any endogenous compounds, baseline adjustment should be clearly specified, as well as, in some cases, the determination of the actual dose that will be used in the calculation of the PK parameters. For example, some drugs are administered per unit body weight, and for other drugs, there may be salt or molar corrections needed. The SAP should also contain a clear definition of the PK analysis set, together with any exclusions clearly specified. It is very easy to overcomplicate this in the SAP or to create problems with a given definition. The PK analysis set should also be defined in the protocol, so if you have a chance to review this, I would highly recommend it. The general considerations are as follows. Firstly, all PK data is usually listed, regardless of subsequent inclusion in summary statistics or statistical analyses. Secondly, any reasons for exclusion should be pre-specified in the SAP and may result in the omission of data from the statistical analyses. And finally, in some studies or for some clients, the samples from subjects randomized to placebo may also actually be analyzed and required to be listed. The analysis of placebo samples is usually stated in the protocol, but can also be confirmed with the bioanalytical lab via the project manager, and should also be stated in the data transfer agreement. But this essentially means that there are two distinct populations for the PK data. Firstly, all subjects who have any PK data at all, which is the basis for listings. And secondly, all subjects who are included in any descriptive or statistical analysis, taking into account any exclusions. So it is important to ensure that any definition of the PK analysis set allows for presentation of all relevant data and does not exclude any subjects with data that may be included in the listings. Any potential exclusions from the PK analysis, and by that I mean the descriptive statistics and statistical analyses, should be documented in the SAP and specified up front. And these can be divided into exclusions that result from protocol deviations and exclusions that may arise due to other issues. In terms of protocol deviations, these could include prohibited concomitant medications, non-compliance with dosing or incomplete dosing, or sampling issues, for example, missed or incomplete urine samples. For exclusions not related to protocol deviations, these may include adverse events, which may impact on the PK data, such as vomiting, or early withdrawal from the study, for example, which is not in itself a protocol deviation, but which may mean that the collection of PK data is not considered sufficiently complete to determine the required PK parameters. In addition, most SOPs that I have seen across several different CROs and pharma companies also allow the PK scientists to exclude additional data at their discretion if these seem to be anomalous and there is good scientific reason to do so. It should just be ensured that this is documented appropriately and that there is a mechanism in place that allows these exclusions to be communicated back to statistical programming, such that the data can be excluded programmatically during the production of the TFLs. In terms of the actual statistical analysis, the description should be largely similar to that for any efficacy or other stats analysis included in the SAP. And this would include a clear and unambiguous stating of the analysis set, the data to be included, the SAS procedure, terms in the model, and parameters to be analysed, as well as clearly stating any PK parameters to be log transformed prior to analysis. In relation to the point about the data to be included in the model, please note that there are some specific rules in place for bioequivalence studies. For example, in a 3-period study with 2 reference products and 1 test product, EMA require that the statistical analysis is conducted with the exclusion of the data not relevant for the specific comparison. Similarly, for a 4-period study where bioequivalence is being assessed in fed and fasting states, the evaluation in the fed state should only include the data from the 2 fed periods, and the data from the fasting periods should be dropped from the input dataset. And these details should be included in the SAP to ensure that any data going into the model is clearly subset as required. There are numerous regulatory guidelines available that cover the entire spectrum of clinical pharmacology studies. Guidelines for bioequivalent studies are both region and compound specific. There are also published guidance documents available for drug-drug interaction, biosimilars, food effects and thorough QTT studies, as well as studies in special populations such as renal or hepatic impairment. It's extremely important to consult the relevant guidance for a specific study type at the design stage in order to ensure that the study is being performed according to the specific requirements. As Louise mentioned at the start, I spent just over 10 years working in early phase biostatistics, and during that time I was involved in many different bioequivalent studies. And the one thing that I did learn was that they're definitely not all the same. Formal bioequivalent studies have different requirements depending on the regulatory body for submission and the compound. One useful website that I have come across is this one, which has all the guidelines in one place, provided of course that you can recognise the flags. I have found this to be updated regularly, although I would still recommend visiting the specific website just to check that there is no later information available. So just to give you an idea of some of the subtle differences between the guidelines, I've included some comparisons based on key requirements for bioequivalent studies. So I guess the first question would be, which PK parameters should actually be included in the statistical analysis? And this is for a single dose study. So if I look at Health Canada, they stipulate that we should analyse Cmax and AUC 0 to T. That's similar for EMA and also for Japan. But then if we look at FDA, on the other hand, they also require a statistical analysis of AUC to infinity. If I ask the next question, which is probably, what are the bioequivalence limits? If I start with Health Canada again, here the limits are stated to be 80.0 to 125.0, and rounding is not mentioned in the guideline. Japan have limits of 0.8 to 1.25, and again, rounding is not mentioned in the guideline. However, if we look at FDA, here the limits are specified to be 80 to 125%, but here it is specifically mentioned that rounding should not be performed. And in contrast, if we look at the EMA guidelines, they specify limits of 80.00 to 125.00, but specifically require this time for this to be assessed after rounding to 2 decimal places. And what this means, of course, is that you can have a drug that appears to pass bioequivalence for EMA and fail for FDA based on rounding alone. And finally, I had a look at the guidance for the other PK parameters that are required to be reported as part of the submission. And here again, there are some key differences. If I start with EMA this time, as a minimum, they require the primary PK parameters, as well as Tmax, the points used for lambda, and the extrapolated area. FDA specifies that the half-life and lambda Z should also be reported. Health Canada goes one step further and requests that the individual ratios of AUC 0 to T to AUC 0 to infinity are provided, as well as, when time to onset is important, the area under the curve to the Tmax of the reference product. And finally, for Japan, they also require the mean residence time, the R-squared, and the individual ratios of test versus reference for the primary PK parameters. And I would also like to add at this point that failure to include all the required information may well result in an application being returned in order to provide the missing variables or other missing data which may have been specified in the guidance. So the previous slides are certainly not meant to be an exhaustive list of differences, but were included just to provide an insight into why it is important to consult the specific guidance before starting the study and when writing the SAP. Guidance, with whatever study type, provides recommendations on several key areas from study design through to analysis, so it is always important to consult the relevant website for the latest guidance to ensure that the data that you provide meets the minimum submission requirements for the appropriate regulatory body. So the next section of my presentation is going to look at common data issues and protocol deviations. In my experience, PK studies often present with many similar and recurring data issues, so I thought it would be useful to highlight some of these in order to facilitate review of data and how these may influence the handling of specific data during analysis. The most common protocol deviations are time window deviations for collecting blood and potentially urine samples. Typically, some protocols may allow for a specific window around the planned collection times, and this is advantageous because otherwise we end up with hundreds of sampling deviations, most of which are really very minor. So, for example, a planned sampling time of 2 hours post-dose may have a plus-minus 5-minute window, which means that samples taken within 5 minutes either side of this are not considered as protocol deviations. I would really advocate this approach because such small deviations will not have an important effect on the calculation of the PK parameters, and any listing of protocol deviations is then focused on those sampling time deviations which are actually of significant magnitude. I would also add at this point that reliance on clinic or monitor reporting of time window deviations is, in my experience, incredibly problematic, as this results in a lot of missed deviations or incorrectly stated deviations due to typos or miscalculations. it is far better to designate these as programmable deviations and actually program them based on the actual sampling times from the database. Secondly, missed PK samples may also occur during the study. This may be for a range of reasons, the simplest of which may be that the sample was just missed in error, or perhaps there was an issue with the cannula, which meant it wasn't possible to obtain the sample. I'm going to talk more about urine collections on the next slide, so I'll skip over this one for now. As for any study, administration of prohibited concomitant medications is a common issue and may be of potential importance from a drug interaction perspective. It can therefore be useful to provide a list of concomitant medications to the PK scientist in order to determine if any of these are considered to have an impact on the analysis. Dosing deviations are extremely important, so it is necessary to ensure that the deviation contains sufficient information to evaluate the potential effect. For example, if the dose is being given via infusion, it is important to document the volume that was actually administered in the case that this was incomplete. And for an infusion that was interrupted, it is important to document when it was interrupted and how long it was interrupted for. Finally, there may be other study-specific deviations. For example, in a food effects study, the failure to complete the high-fat breakfast would be a valid reason for exclusion. Urine PK data is particularly challenging. Imagine that we have urine volume data for a particular subject with data from various sampling intervals to 48 hours post-dose. We should always be extremely suspicious of zero urine volumes, especially in a collection lasting for 12 hours. One of the most common reasons for this that I have seen is subject voided into the toilet by mistake. In other words, urine was excreted, which therefore probably also means that some drug was excreted, but this was not collected in error. I've also seen many other comments, such as subject had 2 containers and there was no urine in the second container. And here a mistake can be made in that we should sum the contents from the 2 containers, but somehow the first one got missed and then 0 was entered. I've also seen similar deviations for spilled containers or even swapped containers with another subject. It is very important to distinguish between a true zero and a data issue. I would generally take the approach of querying all the 0 volumes, although these could be corrected at early time points when urine collection intervals may only be of 2 hours duration and the subject was not able to produce a sample. But it is really important that the site doesn't enter 0 when actually we don't know it was 0. And this is because this will genuinely get used in the calculation of the amount excreted and will result in an overall underestimation of the cumulative amount of drug excreted. In terms of the analysis, any invalid urine collection data should be excluded from the specific time point and all subsequent time points. So in this example, the amount excreted from time 24 to 36 should not be calculated, assuming that this was a data error, and the cumulative amount excreted should only be calculated up to 24 hours post-dose. Finally, one practical issue to mention about this is regarding the linking of comments and deviations to a specific sample. Often, the way the database is set up, sample-level deviations are not possible to be linked to the specific record, or comments are provided as free text in a comments field, and it is not possible to programmatically extract the relevant information. Thus, it is important to think about how these deviations will be reported and to make them standardized and consistent such that exclusions could be handled programmatically. Another thing to look out for is to discourage any very late collection of samples or collection of samples at the same time. I've seen instances where a site forgot to take 1 sample and then took 2 samples at the next time point in order to try and ensure that all samples were taken per protocol. And this is shown in the example here, where the 6-hour scheduled collection was forgotten and then taken together with the 8 hours post-dose sample. This firstly rather defeats the purpose of obtaining a profile over time, But it should also be noted that WinNonLin cannot handle 2 PK samples with the same date and time, so we would anyway have to delete one of these from the analysis. Another issue that I have encountered with dates and times is regarding the entering of the dates, although I think more recently this has been less of an issue as full dates and times may be captured automatically. The example here shows a PK profile for blood sampling up to 24 hours post-dose. And as you can see at the 16-hour time point, the calendar day rolls over to the 2nd of August. However, if the date is not collected for every sample, what happens is that the entire set of samples is associated with the first date on the CRS page, and therefore it will result in the 16-hour sample looking like it was taken prior to dosing. Similarly, for urine PK data, it is important to collect the date for both the start and the end time of the collection. In the example shown here, we are collecting the date and then the start and the end time for each collection, which actually looks perfectly acceptable. However, when this data is mapped to SDTM, the same date will be associated with both samples on the row, such that when the map data are received, it would appear as though the end of the collection was prior to the start of the collection for the 12 to 24-hour collection, and for the 24 to 48-hour interval, it would appear as though it had a duration of 0. So if you are ever reviewing a CRS for a PK study, it is important to ensure that dates are being collected for each and every sample for blood sampling and for start and end of every collection for urine sampling. So the final point I want to make about actual sampling times was based on a lesson very well learned for me, and this is clock changes due to daylight savings. In the example here, we know that the clocks went back 1 hour at 2 AM on the 28th of October, and of course we all celebrated with an extra hour in bed. Here, at the 24-hour time point, the actual clock date and time was entered, and for this subject was 0658, and for this subject was 0700. And these are both local times. If these values are then used in the calculation of actual sampling time, these will result in both samples appearing to have been taken 1 hour early. Thus, when calculating the actual relative sampling time, it is important to make the same adjustment such that the time relative to dosing is calculated correctly. If you're reviewing a listing of PK concentrations or time window deviations, if you do see multiple deviations of exactly an hour at the 24-hour time point, this should definitely lead to some sort of suspicion. And of course, the same issue will take place when the clocks go forward, and in this case, it will appear as though samples were taken at 25 hours after dosing. So in terms of lessons learned, I think that there are 2 key points here. Firstly, we need to understand how the database is set up in terms of daylight savings and whether automatic adjustments are being made and whether times are being captured as local times. Secondly, we need to ensure that when we're calculating actual sampling times, any necessary adjustments are being taken into account. One pragmatic thing to note, however, is that while 1 hour out of 24 hours may be considered important, if you have a drug with a long half-life and samples that are going out for several days, the relative importance of daylight savings decreases. So, for example, a deviation of 1 hour for a time point of 168 hours post-dose would make very little difference to any calculation of PK parameters. The final section of the presentation is going to discuss logistical considerations for PK studies, especially those adopting C-disk structures for SDTM and ADAM datasets. I've split this section of the presentation into 2 process flows, firstly for the PK concentrations, and the next slide for the PK parameters. It's really important to understand the process to ensure that the study runs smoothly and everybody knows what to expect and when, so that timelines can be planned accordingly. And this is a really key part of the successful project management of any PK study, especially where several teams are involved, which may include the clinic, those responsible for sample shipment, the bioanalytical lab, data management, biostatistics, and of course the PK scientist. So the first part of the process, once the samples have been obtained and shipped, is the receipt of the concentration data from the analysing lab. This is then combined with the CRF data into the SDTM dataset. The dosing data is then merged with the PK sampling data to create ADPC, and from here the input file for Win-non-Lin can be created, which is usually in a CSV format, and this is then sent to the PK scientist. It is very important when creating Win-non-Lin input files, if these are being done in biostatistics, to determine the exact requirements from the PK scientist. Whereas ADPC typically contains all the concentration data, including plasma and urine data, the calculation of PK parameters usually requires a splitting of these files, potentially into those for single dose profiles, those for steady state, and typically the urine data is kept separately, as these do not need to be modelled in Win-non-Lin. Thus, as part of the planning, it is very important, if biostatistics are responsible for creating the input files, to ascertain the exact requirements and ideally provide a test file in order to help the process run smoothly. It should be mentioned, however, that this is not the only way of implementing this process, and I have worked in several different ways depending on the requirements or SOPs being followed. Sometimes the PK scientist may use the SDTM datasets directly. Alternatively, the PK scientist may receive the ADPC dataset, and any further data manipulation, for example subsetting the file, may be done by the PK scientist. And I've worked on studies where each of these approaches has been used. I wouldn't recommend the first approach, however, whereby the SDTM datasets are sent directly to the PK scientist, because then it means that the actual sampling times are being calculated separately outside the ADPC domain, which may lead to discrepancies versus the ADAM dataset. And I've also added a reminder here regarding urine PK data, which should also be transferred to the PK scientist if the urine PK parameters are being calculated outside biostatistics. So in terms of the PK parameter process, following the calculation, the relevant PK parameter data should be returned, usually again in CSV format, and this is incorporated into the PP domain and can then be used to create ADPP, from which the PK parameter TFLs are programmed. One of the most problematic areas in the entire process is the decision to exclude certain PK parameters or even PK concentrations from the analysis by the PK scientist. For example, the PK scientist may determine that one of the concentrations was anomalous and should therefore not be included in the calculation of the PK parameters. Similarly, the PK scientist may decide that the calculation of the AUC infinity was not technically accurate and should therefore be excluded. So how does this information get returned to programming? Flagging of PK parameters is the easiest to handle. Win-non-Lin allows export of the final PK parameters in a vertical structure, such that a flag and standardized comments can be added at the parameter level for a given subject. These flags can then be mapped into the supplemental PP dataset and incorporated into ADPP accordingly. And this will enable certain parameters to be flagged in the data listings and programmatically excluded from any statistical analysis. For PK concentrations, this is slightly more tricky. The easiest way to flag any excluded concentrations is for this to be done in the Win-non-Lin input file, and this file then returned to biostatistics. And this can then be merged back with ADPC so that the flags can be incorporated and used to exclude the required samples from any analyses. However, this does mean that we are then receiving an external source file rather than the SDTM being the source of all the ADAM data, so this should be documented accordingly and explained in the ADAM Reviewer's Guide. In my experience, this area is probably the most problematic in the analysis and reporting of any PK study, so it is well worth investing time in discussing the process, agreeing on how it will be performed, and documenting accordingly. I have found it very worthwhile when working with new clients to understand whether they have any expectations in this regard and what their usual process is, and to be able to recommend a process. So one final area relating to the overall PK process is the requirement to perform a dry run. And I think a dry run is now fairly commonplace across all studies and phases to allow review of the format and intended layout of the final TFLs, and to allow pre-programming with the aim of reducing timelines after database lock. The problem with PK studies is that the data itself may be considered unblinding and may not be available until after database lock. So what are the options? Well, we can create dummy data for both PK concentrations and PK parameters, but how do we know how similar this will be to the final data? Typically, we base rules for rounding in tables on the source data, and we format figures based on the observed range of concentrations. So if we don't have very representative dummy data, this can result in a significant degree of program modification after database lock when the real data is obtained. It is also very useful to consider that it is not only the programs for the TFLs that need to be created, but also any other programs used to create SCTM datasets, ADAM datasets, and also the Win non-lint input file. Thus, it might be advantageous to try and mimic the entire end-to-end process as part of the dry run. So in an ideal world, what does the dry run look like? The bioanalytical lab itself would provide the dummy concentration data according to the data transfer specifications, and this would then be sent to data management or biostatistics for creation of PC, and then we would calculate the PK parameters, return them to biostatistics, and produce the TFLs. The problems with this are several. Firstly, the analysing lab may not agree to provide dummy data, or there may be a charge for it. And secondly, sometimes it's not always known what we mean by dummy data. And I have sometimes waited a long time to receive dummy data from a lab, and when it's arrived, it only contained one subject, and the same concentration was included at every time point, which clearly doesn't work well for statistical analyses and certainly doesn't work well for the calculation of PK parameters. Similarly, if the calculation of PK parameters has been outsourced, there may be an additional charge to run the analysis on dummy data for the dry run, and of course they'll only be able to run it if they've received appropriate dummy data. One halfway option is to create dummy concentrations and dummy PK parameters separately in order to allow production of datasets and TFLs, but this does have the disadvantage that it does not mimic the end-to-end process, and also means that the PK parameters and the concentrations are not aligned. And what I mean by this is, if you simulate them separately, your maximum concentration observed in your dummy concentrations doesn't match your Cmax in your PK parameters, and this can cause confusion when reviewing the dry run TFLs. There are many ways to go out performing a successful dry run on PK data, but again, the logistics should be discussed and agreed at the outset, and the timelines planned accordingly. So in summary, PK is a key component of many early phase and clinical pharmacology studies. Challenges relate to logistical and practical considerations, especially with regard to data flow and dry run requirements. There are some common data issues and protocol deviations to be aware of, and if performing a study for regulatory submission, it is important to consider the relevant regulatory body guidance at the outset. Thank you all for attending, and I will be happy to answer any questions.
Thanks very much, Claire. So we have received one question whilst you were presenting. Just to recap, for anybody who does want to ask Claire a question, they can do that via the questions box on your toolbar. You just need to type in the question and I can present that to Claire. So in the meantime, Claire, I do have one question. Do you have any advice, guidance on performing interim PK analyses for dose escalation studies?
Yeah, so for dose escalation, typically we analyse the PK data in between each cohort in order to help to choose the dose level for the subsequent cohorts. But obviously one of the main issues with this is that the data are considered unblinding. So firstly, we need to agree that the analysing lab provides the data using alias subject numbers. So they would receive a copy of the randomisation code, and then they would assign, dummy numbers to the subjects before they send the data back. Now, typically, we don't calculate actual sampling times for dose escalation studies, so the entire process can actually be done independently of biostatistics, and it could be done just between the lab and the PK scientist. But one thing to mention is that when producing any tables of data for dose escalation studies, we should always make sure that we don't show the N for the descriptive statistics. And this is because if you've got a subject, for example, that has either dropped out or failed to provide a sample, if we show the N at each time point or the N for the PK parameters, it will be very clear whether or not the subject that dropped out was on active or placebo. So the main kind of thing to know is about preserving the blind, and if you are producing tables, figures and listings, just to make sure that we don't show the number of subjects in the analyses.
That's great, thanks Claire. So we don't have any other questions that have come in throughout the session. But what I will say though is that we will be sending a copy of the slides to all of the attendees, and we've been recording the session as well. So if you did want to go back and listen to any specific parts, you'd be able to do that too. So without any further ado, we'll leave you to the rest of your afternoon. Thanks so much, Claire.
Thank you.
Thanks.